Okay, in Chapter 8, we're going to be looking at the cellular basis of reproduction and inheritance. We're going to be looking at cell division and reproduction. We'll be taking a look at the eukaryotic cell cycle as it applies to, or as it involves, mitosis. And then we'll also be looking at the eukaryotic cell cycle involving meiosis. And then we'll be, at the end of the chapter, we'll get a little bit into chromosomal number. And now, in the case of cell division, we want to keep in mind that it plays a vital role certainly in living organisms. In some cases, organisms are using cell division to produce offspring that are genetically identical to themselves, and that's an example of asexual reproduction. So in the case of asexual reproduction, gametes are not involved, and what happens is that the daughter cells, which is going to be a clone, will be genetically identical to the parent. Now that works perfectly fine when the environment is certainly conducive to that set or mixture of genes, and that is certainly the case sometimes for like yeast cells. So in this diagram here, we have a yeast cell, and it has duplicated its chromosomes, and now it's budding. It's producing a clone of itself, which will be genetically identical to itself. The C star, on the other hand, is here. There's been a C star that has fragmented, and it's beginning to regrow its arms. And so that's another example of really one C star that probably has fragmented and is actually now going to be a couple of clones of that one C star. And in this case, we see that an African violet, a cutting from an African violet, has been planted, and there's a whole new plant going to grow off of that cutting. So this is a way in which clones can be produced by way of asexual reproduction. Now, in sexual reproduction, of course, the offspring are going to be genetically distinct from either parent, and that really is the upside to sexual reproduction. So in the case of humans, whenever there is fusion of gametes from two different parents, then the mixture of parental genes in that offspring means the offspring will be different from either parent. And of course, what that zygote, or that fertilized egg, will do is carry out mitosis, which means one cell becomes two identical cells become four identical cells, and so on until an embryo develops. And so those cells will all be like cells. But in time, some of the cells, as they continue to divide, will differentiate, become different types of cells, even though they contain DNA that's all the same. And so eventually a human will develop, and that human will consist of trillions of cells, and those almost all trillions of cells will have a full complement, a full set of the same DNA, the same genes. Now, sexually reproducing organisms can also, though, use cell division for replacing tissues. For instance, we're constantly sloughing off skin cells, so to renew our skin, we can replace them by way of cell division, or even for growth, as we watch our children grow from being small to being full-size adults. There's an awful lot of cell division certainly going on in that process. And so millions of cells are literally dividing every second to replace cells that have been lost or that have been damaged. Now, in the case of prokaryotes, like bacteria, they can reproduce by binary fission. And what they will have is oftentimes a circular, single circular chromosome, DNA strand. And what they'll do is they'll make a copy of that single circular DNA. And here we see that in this second drawing where it has been copied. And also notice that the cell is beginning to elongate a bit. That's another step that they will carry out at this time. And then, in time, the plasma membrane and the cell wall begin separating the one cell into two cells. And so what happens is ultimately is the parent cell will divide into two daughter cells, and this again is asexual. Now in the case of the eukaryotic cell cycle in mitosis, what we have is we have chromosomes that must be duplicated before a cell can divide. And most human somatic cells, in other words, these are non-gametic cells, carry 46 chromosomes. Dog somatic cells carry 78. Mosquito somatic cells carry 6 chromosomes. Now, within most all of our somatic cells, on those 46 chromosomes, we'll have just under 21,000 genes. If we were to take our 46 chromosomes, stretch them out, and tie them end to end, they would measure out about 2 meters in length. Now, when we speak of a chromosome, we're talking about both a DNA molecule and its attached proteins. From time to time, we'll use the term chromatin. Chromatin is when the chromosome is really in its thin, diffuse state. In other words, it's not condensed. Another thing we need to keep in mind is that a DNA molecule consists of two complementary strands of DNA. In other words, on one strand, you'll have, say, an adenine type nucleotide, and on the other strand, directly across, you'll have a thymine. On one strand, you'll have a guanine. Directly across, on the other strand, you'll have a cytosine. So you have this complementary base pairing between the two strands that make up one DNA molecule. Now, prior to let's do a little count here of chromosomes, DNA molecules, and strands for mosquitoes and humans. Now, how many chromosomes did we say just a bit ago there are in, say, mosquitoes in their somatic cells? 6. For humans, we said 46. What about DNA molecules, though? How many DNA molecules do we find within a mosquito somatic cell? Well, remember, if we look over here, here is one chromosome. That one chromosome consists of one DNA molecule. And that one DNA molecule consists of two strands. So that's what we have over here on the right side of the diagram. Now, so we go back to our question. How many DNA molecules in a mosquito? It's certainly going to be 6. How many DNA molecules in a human somatic cell? Still going to be 46. But what about strands now? Well, we've already looked over here and we've noted that 2 strands make up one DNA molecule. 2 strands make up one, I'm going to call it a non-duplicated, non-replicated chromosome. Okay? So now the question, how many strands in the nucleus of a mosquito? Of course, it's going to have to be 12. 2 strands per chromosome, 2 strands per DNA molecule. So that means it would be 92 for the human. Now, prior to dividing, a cell will duplicate its chromatin. So here, what we're going to look at over here is here's a non-duplicated chromosome, a chromosome that has not been duplicated yet, consisting of one DNA molecule, 2 DNA strands. But what's going to happen is that non-duplicated chromosome will be duplicated. And at this point, we refer to this as a duplicated chromosome, consisting of 2 sister chromatids. Now, each sister chromatid, this left sister chromatid, consists of this DNA molecule, these 2 strands. This right sister chromatid consists of this DNA molecule, these 2 strands. So these 2 together make up one duplicated chromosome. And we do that to all 46 chromosomes. So we end up duplicating our chromosomes. We do that in what's called the S part of interphase. So once again, a duplicated chromosome consists of 2 sister chromatids. Now, in this electron micrograph, we see here, this is a left sister chromatid, and this is a right sister chromatid. Now, they should be genetically identical to one another. And one of the things you notice is that the sister chromatids look like they're kind of stuck together a bit. And that's because there are cohesin proteins that actually help hold them together. Now, another term we want to point out is centromere. Centromere is where the chromosome constricts a bit. It's kind of like the waist of the chromosome. That's the centromere. Now, as we've said before, each chromosome is a duplicated chromosome. It consists of 2 DNA molecules, 4 DNA strands. So let's go back to what we were talking about earlier, except now we're looking at the configuration after the chromosomes have been duplicated. So in this state here, where each chromosome consists of 2 sister chromatids. So how many chromosomes would you find in a mosquito somatic cell that has duplicated its chromosomes? You'd still say 6. There are 6 duplicated chromosomes consisting of 2 sister chromatids each. In human, it'd still be 46. What about the number of DNA molecules? Well, think about that. Each chromatid, and there are 2 chromatids per chromosome, each chromatid consists of 1 DNA molecule. So that means there are 2 sister chromatids, there will be 2 DNA molecules. So for mosquitoes now, that means there will be 12 DNA molecules in total. For humans, it's going to be 92. What about DNA strands per duplicated chromosome? Well, a duplicated chromosome is 2 sister chromatids, 2 DNA molecules, 4 strands. So, for the mosquito, that's 24. For the human, that's 184 strands. Now, what's going to happen in time as we go through cell division is the chromatids, the sister chromatids, will separate. So one chromatid will go into one cell, the other chromatid will go into the second cell. And what we will end up with then are individual, non-duplicated. Okay, so now we're going to start looking at the cell cycle. And the cell cycle includes both cell growth and also phases when the cell is preparing and going on and dividing. Now, this process we're about to get into has enabled each one of us to grow from a single-celled zygote inside mom to a some 10 trillion celled individual. Now, if we look down here at the drawing for this cell cycle, we see that interphase is a major part of the cell cycle. A cell will spend most of its time in an interphase state. Then, if the cell has pretty well been signaled and been given kind of the thumbs up to divide, it can then start to duplicate its proteins and organelles. It can then duplicate its DNA and complete duplication of proteins and organelles, and this is all in the latter part of interphase. We divide interphase into a G1, S, and a G2. So once again, most of the cell cycle is consumed by interphase. About 90% of the life of a cell is spent in interphase. Now in G1 of interphase, that's when the cell begins doubling most of its cytoplasm. In the S part of interphase, that's when the cell duplicates its chromosomes. Okay, so we will have a whole chapter really looking at the S part of interphase. So oftentimes we'll refer to this S phase or S part of interphase as being DNA synthesis or DNA replication. That is what's kind of illustrated over here. This chromosome that's a non-duplicated becoming a duplicated chromosome consisting of 2 sister chromatids. S phase is where that happens. Now, once it's completed, each chromosome will consist of 2 sister chromatids. And then G2 is when the cell is pretty much finishing up duplicating. Now, we can look at our pie chart again. Once a cell finishes up G2, it can get into the mitotic phase. Now, the mitotic phase is broken down into mitosis and cytokinesis. And I will tell you that strictly, mitosis is really nuclear division of the nucleus. And cytokinesis is division of the cytoplasm. That's when a cell either pinches in half in the case of an animal cell, or a cell plate forms like in the case of a plant cell. And so there's division of the cytoplasm. So Once G2 of interphase is completed, then the cell can move on into the mitotic phase, which once again is divided into both mitosis and cytokinesis. Now, the first phase of mitosis is prophase, and that's what's being shown down here. And in the case of prophase, look at the difference between the nucleus here in prophase and the nucleus in interphase. You can start to see that the chromosomes have condensed. They're duplicated. You can see the two sister chromatids making up each one of them. But what we have early on in prophase is the chromatin fibers condensing into discrete chromosomes. Another thing that's happening is these microtubules are being built out from these centrosomes containing centrioles in the case of animal cells. So these microtubules being built out make up what's called the mitotic spindle. So we have that being done during prophase as well. Now, the mitotic spindle, as I've mentioned, consists of both microtubules and proteins. Another key feature of prophase is that the centrosomes containing centrioles, they begin moving poleward, so they're moving away from one another. We see over here in interphase, they're together. But during prophase, they start moving poleward. They start moving away from one another. And that's another key component of prophase as well. Now, the next phase after prophase is prometaphase. Now, one of the key features of prometaphase, and we can see it here, and that is fragmentation of the nuclear envelope. That is a key characteristic of prometaphase. Also, we see down here, for instance, this microtubule is attaching to this little black button on a sister chromatid, and that little black button is called a kinetochore. That's the kinetochore. So microtubules begin attaching to kinetochores. Now, the kinetochore on, let's say, the south side or on the bottom side of this duplicated chromosome, that kinetochore will attach to a microtubule coming from the bottom centrosome. The kinetochore on the top side sister chromatid will attach to a microtubule coming from the top side centrosome. So we have microtubules beginning to attach to kinetochores of sister chromatids. Now, those microtubules that are attached to kinetochores are just referred to simply as kinetochore microtubules. So now we also have these opposite poled microtubules getting into kind of a tug of war. And so one can actually see chromosomes being jerked back and forth during this tug of war here in prometaphase. All right, now following prometaphase is metaphase. And in metaphase, what we have is, once again, that continued tug of war resulting in a stalemate, and the chromosomes will be aligned along the metaphase plate, and that's what we see going on here. Now, notice that the centromeres are aligned along the metaphase plate, but the chromosomal arms themselves, the sister chromatid tips, may actually dangle off a bit. But in metaphase, the goal is to get everything lined up along. Now, following metaphase is anaphase. Now, in anaphase, what's going to happen is that these little black dots on each sister chromatid represent a kinetochore, which is a protein. And inside each kinetochore is a motor protein. So inside this kinetochore is a motor protein. Inside this kinetochore is a motor protein. And these motor proteins, they're kind of like Pac-Man. They chomp their way along the microtubule to which they're attached. And so this sister chromatid's motor protein is chomping its way along on this microtubule. The motor protein in this kinetochore is chomping its way along this microtubule. So we get one sister chromatid, in this case, moving towards the top centrosome, and this one moving towards the bottom centrosome. So what we get in anaphase that is so critical, we get the motor proteins inside the kinetochores walking the sister chromatid towards the pole it faces. So south-facing kinetochores walk south, north-facing kinetochores walk north, and this leads to separation of sister chromatids of the duplicated chromosomes. Another thing that's going on is that the non-kinetochore microtubules, this microtubule is not attached to a microtubule. I'm sorry, this microtubule is not attached to a kinetochore, and neither is this one. And they push against one another. The endomembrane system is laying down more plasma membrane, so the cell's growing, and these non-kinetochore microtubules push against one another, and that helps elongate the cell so that the cell can be large enough to split in half in due time. So the cells elongate as non-kinetochore microtubules lengthen. Now, the next phase after anaphase is telophase. Telophase is where we have reformation of the nuclear envelope material around the daughter nucleus in one cell, and reformation of the nuclear envelope around the other nucleus, daughter nucleus, that has formed. So that's what we have going on in telophase. So the daughter cells should be genetically identical to one another, and also notice that the chromosomes are beginning to stretch out back into the chromatin form. Now, going on really, along with telophase, we have cytokinesis. Now, in cytokinesis, that's where, in the case of animal cells, you have the cell kind of pinching in half. And this is an actual electron micrograph showing what's called the cleavage furrow forming in an animal cell. So this left half will become one cell, this right half will become another cell. And there's a contracting ring of microfilaments that helps create that cleavage furrow that occurs with cytokinesis. It's a contracting ring of actin and myosin. In the case of plant cells, what they do is they produce this thin line of material that's really from Golgi vesicles, and that is called a cell plate. And that cell plate in time will mature into a new cell wall separating the plant cell right here in this area. So there's some differences certainly between the way animal cells carry out cytokinesis and plant cells. Now, if things go right and well, then plant cells will divide as they should, and replace cells or enable growth and things like that. In studying cell behavior, researchers have noticed that most cells exhibit something called anchorage dependence. Now, in the case of anchorage dependence, what that really refers to is that cells will not begin dividing until they have settled on a substratum. So here we have a culture dish, we have cells inside the culture dish. If we were to rock the culture dish and keep the cells stirred up, floating around in the nutritive medium, they wouldn't divide. But if we place the dish in an incubator, allow the cells to settle to the bottom of the dish, they would be able to sense that they are in contact with the substratum. Their cell surface proteins, those would be transmembrane proteins in the plasma membrane, would be able to basically signal that attachment has occurred and it's okay to start dividing. Now, they have found that some cancerous cells don't require this anchorage. They don't have what's called anchorage dependence. Now, also, most cells, when they do grow, they will fill in the available space, and then they'll stop growing. Once again, they have these cell surface proteins that's able to signal to the cell when things are getting crowded, when they have a neighbor. So crowded cells that are normal will stop dividing at this point. Once again, they're using these cell surface proteins to sense that all available space has been taken up. Cancer cells will continue to divide. It doesn't matter that it's crowded. They will continue to divide. Researchers have also found that there are protein growth factors that can stimulate cells to divide. They've discovered more than 50 different growth factors that actually stimulate cell division. Now, different types of cells are going to respond to certain types of growth factors, or even a combination of growth factors. Now, one growth factor that they have identified is called PDGF. PDGF is platelet-derived growth factor. So let's say that you have injury to your skin, and platelets arrive at this injury to plug up the bleeding. The platelets will begin producing and secreting PDGF. And the PDGF, this growth factor, will actually begin binding to receptors on cells called fibroblasts. Now, fibroblasts are cells that are really common in connective tissue. And whenever the PDGF begins its binding with the fibroblast, that communicates to the fibroblast to start to divide to really heal the wound. Another growth factor that they've identified is the VEGF. That's the vascular endothelial growth factor. And this is a growth factor that actually stimulates the growth of new blood vessels. So, as a fetus is developing or after an injury, then VEGF will be secreted to begin the healing process or the growth process with the building of new blood vessels. What they have found with some cancers is that there is overproduction of this growth factor, and that leads to proliferation of vessels, which supports the energetic demands of the cancer cells, so that the cancer cells can continue dividing. Now, growth factors do once again signal the cell cycle control system. Now, we haven't really talked about the control system yet, but as we've said, most cells are not going to divide unless they've been given kind of a thumbs up, unless they have received a signal to go ahead and do so, and that signal will come from some other cells. So growth factors are indeed the main signals for cell division. Now, there are also intracellular signals that are detected by the control system, and this tells the control system whether key cellular processes are where they need to be, that they've been completed, and whether the cell cycle should proceed past a particular point. So the control system is receiving growth factors. There are also some intracellular signals, and also receives messages from outside the cell indicating the general environmental conditions surrounding the cell, and also the presence of certain signal molecules from other cells. Now, some of the key checkpoints, some of the that are part of the cell cycle control system would be the G1 checkpoint, the G2 checkpoint, and the M checkpoint. So these checkpoints have to receive a thumbs up so that a cell will go ahead and divide. And what's being shown over here is a growth factor produced by some other cell binding to a receptor protein, resulting in conformation change in the receptor, which activates a neighboring protein, which activates another protein's activated. So what we have here is we have signal recognition, or what's called reception, then signal transduction, where protein-protein interactions result in changes in shapes of proteins, and it's this kind of activity that can get a cell past some of these checkpoints anyway, like the G1. So, like we were saying on the previous slide, there are 3 major checkpoints in the cell cycle. The G1 seems to be the most important for most cells anyway. Typically, if a cell receives a thumbs up, a go-ahead signal like a growth factor there for the G1, then the cell will typically go on into S phase of interphase. And remember, that's where a cell is going to replicate its chromosomes. Now, in that, what's going on in that G1 checkpoint is that in the case of, say, the binding of a growth factor, we talked about this on the previous slide. Here we have the illustration of a growth factor binding to a receptor protein. The receptor protein experiences a change in its shape because of reception, which changes the shape of a neighboring protein, which changes the shape of another neighbor, who changes the shape of a neighbor, and so on. So we have reception, we have signal transduction, and that leads to the activation of certain molecules so that the cell knows everything's in pretty good order, and it can go ahead and move on into the S part of interphase. So the binding of the growth factor leads to that signal transduction pathway, the signal transduction pathway consists of protein-to-protein interactions that lead to a thumbs up being given at the G1 checkpoint. And so typically the cell will go on and divide because it has been given the thumbs up there at the G1. But there is a G2 checkpoint and the M checkpoint as well. Now there are some cells like nerve, mature nerve, and muscle cells that never receive a go-ahead signal for the G1 checkpoint. And that's being shown over here in this diagram. You can see that there's this red arrow curving off to the left, and it reads G sub 0. So mature nerve cells and muscle cells, because they're never given the thumbs up at the G1 checkpoint, they will go on into a G sub 0 phase, which is a permanently non-dividing state for those cells. So that's a look at one. But there's also the G2 checkpoint. Now the G2 checkpoint, of course, follows. Let's see, we can go over here maybe. But here's the G2 checkpoint. It is a checkpoint at the end of the G2 of interphase. So the cell has gone through G1, where it's duplicated organelles and proteins. It's gone through S, where it's replicated the chromosomes, and G2, where it has completed essentially the duplication of organelles and proteins. And it's just waiting for the thumbs up to go on into the phases of, say, mitosis or the mitotic phase. So in the G2 checkpoint will involve proteins called cyclin-dependent kinases. We may just call them CDKs, and cyclin. Now, cyclin-dependent kinases, anytime you see kinase, that's an enzyme. Of course, we know it's an enzyme because it ends in ACE. ACE lets you know it's an enzyme. Now, kinases are enzymes that are involved in phosphorylation events. And so cyclin-dependent kinases are enzymes that will phosphorylate other proteins. Now, the way a cyclin-dependent kinase is activated is when it binds to cyclin. And that complex of a CDK plus cyclin is called a maturation promoting factor, an MPF. And what the MPF does is it's really the complex that is involved in the phosphorylation of other proteins. And it leads to the phosphorylation of proteins so that many things can happen, such as, remember in prophase how chromatin begins condensing into chromosomes. Well, the MPF has phosphorylated proteins so that this process certainly gets along the way. And you may remember during prophase we have the building of the mitotic spindle. Remember those microtubules that are emanating from, say, the centrosomes? The MPF is responsible for getting that going. And we also looked at, in prometaphase, fragmentation of the nuclear envelope. Well, the MPF is responsible for activating proteins to get that done as well. Now, the third checkpoint is the M checkpoint. Now, in the case of the M checkpoint, that's a checkpoint in which the cell is going to make sure that all kinetochores, now remember, kinetochores are proteins at the centromeres of the chromosomes, and all kinetochores must be attached to kinetochore microtubules, to spindle microtubules. And the chromosomes, must be lined up there along the metaphase plate. Only then all kinetochores being attached to microtubule chromosomes at the metaphase plate, only then will an appropriate regulatory protein complex become activated. And what that does, that regulatory protein complex being activated, what that does, it sets off a chain of molecular events that leads to the activation of an enzyme called separase. Now, separase, you think of the word separate, and you see ACE, so it's an enzyme. And separase is responsible for cleaving the cohesin proteins that exist between the sister chromatids of the replicated chromosomes. So that is the M checkpoint. Now, we do, from time to time, unfortunately have cells that just grow out of control. And of course, those are cancer cells, which can produce malignant tumors. Cancer claims about 1 in every 5 lives of U.S. citizens. So it's certainly a real threat. And all a cancer cell is it's a normal cell that has undergone something called transformation. It's a normal cell that has become cancerous. It's growing out of control. And the term for that process of a normal cell becoming a cancer cell is transformation. Now, this usually follows a mutation in one or more genes that encodes proteins for the cell cycle control system. And the thing about cancer cells is that they're just not productive contributors to the tissue. The thing that they're really good at doing is dividing, but they're not good at a whole lot else. And oftentimes now cancer cells will be recognized by the immune system as having, DNA that's messed up bad enough or producing weird proteins often enough that the immune system will actually kill off, destroy some of these cancer cells. Of course, we know that what can happen, though, is cancer cells can evade detection by the immune system, and then they can create some real problems. And we see that here in the diagram where there's a tumor that's beginning to develop, and now it's beginning to spread to surrounding tissues. And then it continues to spread, and some of the cells can actually break loose, make their way into the lymphatic system or the cardiovascular system, and then be transported to other parts of the body and begin growing in other parts of the body. Now, once that happens, once the cells break loose and are spread throughout the body, the term for that is metastasis. In other words, we say the cancer has metastasized. So what we have in cancer then is a mass of abnormally growing cells within an otherwise normal tissue. Now, if the cells are remaining at the original site, then that collection of cells is referred to as a benign tumor. Now, a benign tumor, when we say benign, it makes it sound like, well, it's not quite so bad, and that is true. It's not quite so bad. However, a benign tumor can still cause problems if the growth is disruptive to the organ. For instance, like the brain, if the growth is occurring in the brain and disrupting neural activity in the brain. Now, sometimes, though, as the tumor will actually grow out of the original site, spread to neighboring tissues, and then we refer to it as being a malignant tumor. Now, in this case, the malignant tumor can actually displace normal tissue and be disruptive to normal organ function. And so a person with a malignant tumor is said to have cancer. As we described on the previous slide, cancer cells can break loose from their original site, actually move into the lymphatic system and the cardiovascular system, and then spread. So that is a possibility as well. Now, here's another interesting thing about cancer cells. Cancer cells, which are dividing at a rather rapid rate, have high energy demands. So they need to make a lot of ATP, which means they need a good supply of oxygen and nutrients. And they have found that cancer cells may actually signal angiogenesis, which is cancer cells essentially producing signal molecules for blood vessels to proliferate in the area of the cancer growth. Now, cancers are named according to the organ or tissue in which they originated. So carcinomas are really epithelial cells that originate in external or internal body coverings, but it has to do with body coverings. The term carcinoma really means crab in growth. Leukemia, another type of cancer, is if you look up leuco, it would mean white, but it's actually leukemia has to do with then cancer of those white blood cells.